Hello S3, I hope you're all well. This week we're going to start our Nat 5 biology course and we're starting with Unit 1, which is cells. The Nat 5 biology course is split into 3 units. 1. Cells, 2. Multicellular organisms and 3. Life on Earth. We will study these 3 units across the remainder of S3 and S4. The National 5 biology course is assessed with 1 exam paper worth 100 marks and an assignment. The assignment is a practical assessment which you do in school and is worth 20% of the total marks available. During the course, you will be provided with as many resources as we can to aid your learning and revision. For every unit, you will receive a pupil note booklet. This contains all the notes you need for the course. It also contains the course specification and revision tips. You will also get a past paper booklet for each unit of the course. This helps you to structure your revision and identify questions from key areas and key topics that you need to practice. Within this booklet, there will also be command words and what the SQA are looking for, a breakdown of all the past paper questions, telling you which topics, which skills, and how difficult the question is, and also keywords and definitions and key questions, which are really good to help you revise at the end of each subtopic and check you've understood everything you've been taught in class. An additional resource which can be very helpful is Scholar. You can access this through your Go and also using your Scholar login. Just ask your class teacher for this. It contains additional summary notes, animations, practice questions, and end of topic tests. So this week we're going to start with 1.1, which is cell structure. At the start of each lesson, your teacher should provide you with the learning intention and success criteria. This is really helpful in order for you to tick off what you've understood and maybe highlight any areas that you need to ask questions about. They can also be really helpful to structure your revision notes for when you come back to them. So this week we're going to be able to label the organelles found in plants, animal, bacterial and fungal cells. We're going to be able to state their function and describe the differences and similarities between the 4 cell types. At the start of each lesson, we'll also point you in the direction of the course specification. This course specification identifies to you exactly what the SQA can assess you on. It is very helpful when writing revision notes and studying the content, as you know exactly what you need to look at. Sometimes it can be easy to get carried away and learn more than you actually need. So to start off this topic, first I want you to think back to S1 and S2 and S3 when we looked at cell types. Throughout those years we have focused on only plant and animal cells. However, you have learnt the structure and key organelles within them already. So, can you draw and label a plant and animal cell? What structures do each of these contain? And what functions do those structures do? You might also want to try and identify any similarities and differences between the 2 cell types. Take time to think about these questions before moving on. You can pause the video. So now on to cells. There's a short YouTube video in the link here that you can watch. However, we'll go through all the information. So cells are the basic units of life. They come in a variety of shapes and perform many different functions. Organisms made of a single cell are described as being unicellular. Organisms made of many cells, such as animals, are described as being multicellular. So in this unit, we focus on the cell itself, whereas in Unit 2, we look at multicellular organisms. So what we look at is how the cells specialise and work together in their hierarchy in order to allow a multicellular organism to survive. So cells may be classed as animal, plant, fungal or bacterial cells. So these are the four main cell types that you need to learn. Cells are microscopic, so to see their structures, we need to use microscopes. We can see more structures clearly if we use stains to colour specimens before putting them under the microscope. So in S1, you should have looked at onion cells, which are plant cells, and cheek cells, which are animal cells, and you would have learnt the difference between these two cell types. So cells contain structures within them called organelles, which can be seen when the cells are viewed under high magnification. Each organelle carries out a specific function within the cell. The diagrams on this slide show the ultrastructure of an animal and a plant cell, and the next slide shows the ultrastructure of bacterial and fungal cells. You need to use Scholar or Bitesize to copy these diagrams and correctly label the organelles within. For an Act 5, you need to be able to identify certain organelles in animal, plant, bacterial and fungal cells. This includes the cell wall, mitochondrion, chloroplast, cell membrane, cytoplasm, vacuole, nucleus, ribosome, and the plasmids, as indicated in the course specification at the start of this PowerPoint. You should also be able to explain the similarities and differences between the ultrastructure of different cell types and identify these. So for example, they could give you a diagram of an animal cell and a plant cell without any labels and get you to identify which is which, because there are some similarities, but there are some key differences that will allow you to identify one from the other. You also need to know the functions of the structures that make up cells. There's a table on slide 13. You need to copy this into your jotters and use your summary notes or BBC Bitesize to fill this in. You will either fill in the organelle or function for each row. You will also need to tick or cross for each organelle, indicating which organelles each cell type contain. So we've got animal, plant, fungal, bacterial. The nucleus is the first organelle. It is a compartment in the cell where DNA is stored as chromosomes. The function of this organelle is to control activities of the cell. It is found in animal, plant, and fungal cells. The cell membrane can be found in all cell types. It is a selectively permeable double layer of lipid and protein molecules. It controls which substances can enter and leave the cell. The mitochondria are rod-shaped structures found in the cytoplasm. They are the location where aerobic respiration occurs and most of the cell's ATP is produced. Mitochondria are found in animal, plant, and fungal cells. The vacuole is a compartment in cells containing cell sap. The function of this is for water storage and maintenance of turgor within the cell. It is found in plant cells. The cell wall is found in plant, fungal, and bacterial cells. It supports and strengthens the cells. In plant cells, the cell wall is made from cellulose. Chloroplasts are found in plant cells. They are disc-shaped structures in the cytoplasm. It is the location of photosynthesis. Plasmids are small circular chromosomes found in bacterial cells. It contains genes that help cells to function. You look at these in more detail when we come to genetic engineering near the end of the unit. Ribosomes are found in all cell types. This is the location of protein synthesis. The cytoplasm is the location of many chemical reactions. It is a fluid enclosed by the cell membrane and it is found in all cell types. So that's a roundup of all the different organelles, and you could use this video to try and fill in your table if you don't have access to BBC Bitesize. So it's really important in Nat 5 when learning the structures of cells to be able to identify structures on a range of different diagrams. It can be really easy to focus on learning your organelles using these diagrams here and the ones in your summary books, and then you get to the exam or unit test and you see a completely different diagram and it throws you off. So on this slide there's a range of SQA diagrams. Compare these to the version you have drawn in your jotter and check you can identify the key structures. Also check you can identify the cell types from the different diagrams. So that is the knowledge content of 1.1, but we're also going to look at a problem solving skill of the week. Problem solving is a key component of the Nat5 course, with approximately 30% of the exam marks going to skill-based questions. And it's these skill-based questions that most people find difficult in the tests. So this week we're going to focus on converting measurements of lengths to alternative units and also calculating the total magnification. We're also going to look at calculating averages. So in biology, there are numerous units for sizes. This is because there's a huge range of size. You need to be able to convert between the different units. For example, if you were going between millimeters and micrometers, you would have to multiply by 1,000. So you find in biology, especially in this unit of cells, that the main units of measurement that you will find you'll use would be micrometer. So that is the best unit to measure most cells. But sometimes you'll find that things are measured in nanometers or millimeters, and you might be asked to go between them in order to complete your calculation. So the animation on slide 19 will help you to put into perspective the size of a variety of things. So have a look at that. On slide 20, there are some practice questions for you to do to practice converting between millimeters and micrometers. This is really important because there will be several questions in your assignment this week which will involve converting units. The answers are on the next slide for you to check. So when viewing cells under the microscope, you may need to calculate the magnification. You'll be given this equation, but make sure you read through this slide so that you understand. You may also be asked to convert the width of the field of view from millimeters to micrometers. This then allows you to calculate averages. So the most common question you could be asked is one like this. So the diagram shows the field of view down a microscope. You need to calculate the average length and width of cells. To do this, you need to first convert the length to micrometers. So they've given you the field of view in millimeters and you need to convert it to micrometers. So it tells you here that 1 millimeter equals 1,000 micrometers. So 2 millimeters, which is the field of view, length and width, would be 2,000 micrometers. You then need to divide the length by the number of cells across the field of view. So there are 5 cells across the middle of the field of view. That would make the average length of a cell 400 micrometers. If they asked you to do the width, that would be up and down. So it would still be 2 millimeters into micrometers, so 2,000 micrometers, but then you would be dividing it by 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 cells. So there's 10 cells running upwards. So the average width of the cell would be 2,000 micrometers divided by 10, so 200 micrometers. There will be a lot more questions like this on your assignment this week. And then there's a few more here for you to practice. So your assignment this week will be a quiz. The quiz is a combination of key terms and definitions, which you would normally find in your past paper booklet, and past paper questions for 1.1. So the way I've made this form has all the questions embedded in it. There may be some problems with the images loading. Please let me know if this is the case, as it's essential in order to answer the questions. However, there is also a past paper booklet in your files section that you can turn to if everything goes wrong. Make sure you ask your teacher if you're struggling to get access to this assignment fully. So what you have to do during the assignment is use the drop-down menu to choose the correct term to match the definition given in the question. This is the first section of the assignment and it's your key terms for this 1.1 subtopic. There's only one correct answer for each definition. The next section of the assignment is past paper questions, which are a mixture of multiple choice and short answer questions. Throughout learning Nat 5, we will do a lot of past paper questions, as this is key to help you understand how you might be examined for this topic and to check your understanding of the subtopics so far. So that's everything for this week. If you have any questions, make sure that you ask your class teacher and participate in the live sessions where I'm sure they'll be going over many of the things in this PowerPoint.